last lecture, I described how some human viruses are implicated in cancer. I recall I described two ways to show this. First, epidemiology, population studies and case control and cohort. And secondly, finding viral DNA inserted into the host chromosome. Now let's turn to what exactly the viral DNA does to cause cancer when it gets inserted. And also, how is this related to the 90% of cancers? Remember, only 10% are caused by viruses that are not. These 90% are not caused by viruses. This is the field of cancer molecular biology involving DNA and proteins. My story to introduce this topic is the story of a scientific hero. Heroes are hard to find these days with the mass media, but there's truly a scientific hero of the last century, and his name is Jonas Salk. Jonas Salk developed the polio vaccine in 1955. Polio is caused by a virus, and to make the vaccine, Salk needed a lot of virus, and so what he used was poliovirus that had been inactivated. He treated it with a chemical. Salk and later Albert Sabin, who developed the oral live vaccine, needed to grow a lot of poliovirus. And to do this, he took monkey kidney cells that can be infected by poliovirus, brought them into the laboratory, infected them with the virus, and he got a lot of viruses out. So here's his model, which you've seen before. You have the virus, and it's got some DNA, and it shoots it into the cell, and it takes over the cell. And in this case, the monkey kidney cell, get a whole lot of viruses coming out. And so Salk would use, he purified slightly, but he'd use this extract that came out, and then he'd treat it with chemicals, and he'd say, I've inactivated the poliovirus. He'd check to make sure it was inactivated, and then you would give it to children. Everything's fine until 1960. Working at the National Institutes of Health, this is about 5 years after polio vaccine was first used, and it had been given to millions of people. Scientist named Bernice Eddy found some monkey kidney cells that didn't have poliovirus in them at all, died spontaneously. And she thought, why'd they die? Well, she thought maybe there's a virus in them or something that we just don't know about. So she took some of the cells, the extracts of the dead cells that might contain a virus, and she injected them into a laboratory animal. Didn't have a lot of monkeys handy to do it. So she injected it into a standard laboratory animal, which in her lab was hamsters. It's a rodent. And so she injected them into hamsters, and she expected if it was a virus, it might produce a lot of viruses, an infection. No, that's not what happened. They got cancer. So she had unwittingly discovered a tumor virus. Other people isolated that virus. They called it simian virus 40, or SV40, because it was the 40th monkey they were experimenting on. Simian is a monkey word. And they got a test for it, find out if there was simian virus 40 elsewhere. And lo and behold, there was simian virus 40 unwittingly in some of the monkey kidney cells that were used to produce polio vaccine. So millions of children in the 1950s who got Salk vaccine also, in many instances, got SV40 as well. SV40 being a virus that causes cancer in laboratory animals. This set up a massive cohort study. And I'm part of that cohort study because it turns out my blood was tested and I had a lot of antibodies against SV40 after I got the polio vaccine. And so every year or so I get a questionnaire, do you have cancer, how are you doing? And a couple of times I gave blood and they tested the blood, etc. So a huge cohort study. People injected with vaccine that has unwittingly had this tumor virus in animals, and people who didn't. And so far, and I haven't heard from them in a number of years, the epidemiology, there's been papers published, show no effect of SV40 on humans. Thank goodness. Meanwhile, back at, turns out, the Salk Institute. Salk stopped working on the Salk vaccine, and he set up a research institute in Southern California and recruited a really good virologist named Renato Del Becco. Renato Del Becco was working on bacterial viruses. He'd been a pioneer in that field. And he got the idea that maybe SV40 virus could be used just like a bacterial virus and as a model system. If he took rodent cells, hamster cells or rat cells or mouse cells, with no virus in them and added this tumor virus, he could get tumor cells. So this could be a nice model system to study cancer viruses in the laboratory. Now, if this monkey virus infects monkey cells, he gets only a productive infection. They don't get cancer, right? So there's two different hosts. In monkey cells, they give an infection to produce viruses. In rodent cells, they give an infection, but the virus splices into the host and causes cancer. So the question Delbecco asked is, what's going on in these rodent cells where the virus presumably is hanging out? Well, how could he prove that the virus was spliced in? Well, this is, again, remember the alternate lifestyles, only now we're doing it in a tumor situation. So here would be the permissive cell, which would be the monkey cell, and it produces a lot of viruses. This is the non-permissive cell where the virus is hanging out. Delbecco and other people showed by two experimental lines that the viral DNA was indeed hanging out in SV4 in the rodent cells, and it could be reactivated. The first was a really brilliant experiment. Here's the idea. A cell that produces an infection, like a monkey cell, must have some chemicals in it that help the virus produce more viruses. So when a virus infects, the monkey cells have something in it that allows the virus to go ahead and produce more viruses. Of course, the rodent cells don't have it. So he took a rodent cell, he did a cell fusion. He took a rodent cell that didn't have SV40 and doesn't allow SV40 to be produced, and he fused it with a monkey cell that was uninfected. So this is a rodent cell that has SV40 in it, but it's hiding, he thinks it's hiding out, he doesn't know. And he infuses it with an uninfected monkey cell. Now the fused cell is going to have that monkey factor, right? The monkey factor went over, and lo and behold, the rodent cells started producing virus. So this was a terrific experiment because it showed that the virus was latent and was hanging out in the nucleus. The second was the fishing experiment I described last time, where he took a probe, that is something that was complementary, CCCC and GGGG, remember, complementary to the virus, and he looked for SV40 hanging out in the rodent cell, and indeed it was. A rodent cell that had been infected with SV40 was in there. Okay? So conclusion, viral DNA hanging out. It's in there. How does cancer happen in that viral infected cell? Well, Delbecco and others attacked this by the same way that Leland Hartwell, you remember that guy who worked on yeast, attacked the cell cycle. Hartwell had these mutants that were stopped at a certain place in the cell cycle. Delbecco had a mutant SV40 that when it infected cells that would normally be tumor cells, this normal infection, normal mutant viruses normal cells would become cancerous. These are cancer cells. When the virus DNA was spliced into the host and it was mutant virus DNA, the cell just was normal. So now you could compare, right? You could compare the virus that didn't cause cancer with the virus that did cause cancer. And he got the gene that was involved in causing cancer that way. This led to Delbecco receiving, as I mentioned previously in the course, he received the Nobel Prize for this work. So now we have viruses involved with cancer and how they do it at the molecular level. I'm going to describe a number of things in this course, in these next two lectures. And they are, first, that the level of gene expression can be controlled. So we're going to review a little molecular biology. And then we're going to see how tumor viruses and oncogenes use these gene control mechanisms to cause cancer. Then I'll turn to the normal cells. How do normal cells that aren't viral infected actually get cancer if they don't have these extra genes that come in from the tumor virus like SV40 had? In the next lecture, I'll describe the various cancer-causing genes in normal cells, those that control gene expression, cell signaling, and cell death. And then we'll describe how these genes in the normal cell become activated to cause cancer. So first, a review of how gene expression is controlled, because that's the system that tumor viruses are going to manipulate. This is the DNA expression system. DNA here is a gene. And remember, DNA has a terrific length in all the cells, 6 feet of DNA in a little tiny nucleus. Each gene is extremely small, and it might be a couple of thousand nucleotides long, but remember, the genome is 3 billion nucleotides long, so a little gene that's going to be expressed as a little location. And that gene gets copied into an RNA molecule, remember, and that's the one that goes out. Here's the DNA getting copied to RNA, and the RNA then produces a protein. So that's a review of genes and proteins. Now, how are we going to control the expression of a gene? More or less of a given gene going to be exposed in this long system, a little gene. How are we going to control how much of it's going to be expressed? One way to do it would be to have a bunch of copies, right? If we have a long system and this gene is only a little bit, right, one copy of it, if we have 50 copies of it, it's more likely you'll get more of that gene expressed, and you'll see how this happens in cancer. A second way is to control the expression step. In other words, the DNA copied to RNA. So we could promote that, make more of it, or we could make less of it. And a third way is to do things after you make the RNA, what's called post-transcriptionally. So let me look at those 3 ways in a little more detail. First, more copies of a gene. Recall, DNA duplication, this is a graphic you saw before, happens with the original DNA getting copied into 2 strands. And this happens during the cell cycle just before chromosomes get very short and go into cell division. Well, look, there are 6 feet of DNA. And you'd figure, all right, we got chromosomes, maybe each one 100 million units to do. Let's just open it up and start replicating it one by one. So you say, there's a T here, I'll put an A. There's an A here, there's a T, right? And you just go all the way through the chromosome until you reach number 100 million. Unfortunately, our chemistry just can't go that fast. This is all done in a couple of hours. So we need multiple growing points on DNA. So here is a typical human chromosome with multiple growing points. So there might be 1,000 growing points all working at the same time to get all the DNA duplicated during this couple of hours that you have during the cell duplication cycle. Each of those growing points has a name. It's called a replicon. That's not that important. The important thing is you've got multiple growing points, and each one just duplicates once. But sometimes slippage occurs. And if slippage occurs, you'll get more than one copy. It stutters, chemically speaking. And you can get more than one copy and many copies. And so you get an amplified gene amplification. And amplified genes can cause cell division if they are genes that produce proteins important to stimulate dividing cells. We'll see an example of that later on. I said the second way of controlling the expression of a gene would be to control the DNA getting copied to RNA. So here's this little gene in there, and it's getting copied to RNA. How are we going to make it make more expression? More expression. Recall, cell differentiation, right? In cell differentiation, specialized cells, each specialized cell makes a different specialized product. So for example, the red blood cell makes hemoglobin, and the hair cell makes the keratin, right? But the hair cell doesn't make hemoglobin, and the red blood cell doesn't make hair, right? The control spot is just a switch. It's the ignition switch where transcription or copying the DNA begins. And that region is called the promoter. The promoter is a region on DNA where the DNA copying machinery, it's going to copy to RNA, lands and begins at the expression to RNA. This is extremely important, and it's an exquisite control mechanism. Because by manipulating conditions at the promoter, you can make things go faster or slower. Here's an example. You could take a protein here that binds to the promoter, and we know how these proteins bind to the promoter at the 3-dimensional level. You can see DNA here in red, and the protein, which is in blue, binding to it in a highly specific way. Remember, proteins have a 3-dimensional structure, highly specific. And this will be highly specific to a promoter. Are all switches, light switches, are all ignition switches in every appliance you have the same? No, they're not. One switch turns on one thing, another switch turns on another thing. Same thing in life, right? In living things. One promoter turns on one gene, another promoter turns on another gene. So how's this going to happen? Well, for example, we can regulate it to promoter using proteins. I just showed you a protein binding to DNA. So here's the promoter, and we might have what is called a negative regulation, such that a protein called a repressor binds to it and stops the machinery from transcribing it, from making RNA. That's going to turn it off, right? On the other hand, we could have a different protein that's positive regulation that binds to it and says, come on over, come on down, and transcribe this particular one. So you can imagine an activator for the hair gene is in my hair cells, and an activator in cancer cells might be for cell division genes, genes involved in cell reproduction, and genes involved in metastasis and all those other cell functions we talked about. Here's a drawing, actually, of a protein in some detail, and we know all the molecular structure of this binding to DNA. It has a certain shape. This is the protein. You can follow it here, and it's binding, and this is DNA. And there's an exquisite recognition system going on here. And this is so important for turning genes on and off. Whole biotech companies, whole companies are devoted to single types, for example, of these activator and inhibitor molecules, because to manipulate them, you can manipulate what a cell is doing. The third mechanism I want to describe for controlling expression of genes is controlling after transcription. Remember, you make the RNA, right? You make the RNA, and the RNA's been copied now, it's out there, and it's going to be translated into protein, and, okay? But we can manipulate that RNA too, after it's been made. A recent discovery in nature and in humans and in cancer cells as well are small RNAs. It turns out a lot of the genome is making little small RNAs. And their job is to target messenger RNAs, these RNA copies of genes, for either blocking them or promoting them, usually blocking them. These RNAs are very small. Typically, they're about 20 units in length, and a typical large RNA might be 1,000. So the system is that this is the small RNA, and typically it'll bind to a target RNA, it's called a microRNA, and it'll bind to that target and stop it from doing its thing, from making a protein. So that's a third mechanism, and this microRNA is a very hot topic in cancer diagnostics. Different cancers are being found to make different RNAs. Here's a graphic that shows that. These are different microRNAs, and they're made in breast cancer, lung cancer, ovarian, colon, lymph nodes, etc. And so this is a very, very current area of research where people are trying to see which RNAs, these little RNAs made in large numbers in the cell, are targeting certain RNAs for destruction. For example, you might target in a tumor cell a tumor suppressor gene RNA, a tumor suppressor RNA, destroy it, and so the tumor can grow. The phenotype of a cell can also be affected after the protein is made, right? You remember adding phosphate. Remember kinases, adding phosphate, etc., change protein activity. So a major way to do that is to alter the shape of a protein. So the next topic I want to talk about is, so I've talked about the general aspects of gene expression. Now we can look at how oncogenes in viruses cause cancer, and then talk about normal cells. First, let's look at one that you're familiar with already, RB protein. Remember, RB acts at the cell cycle. So SV40 is infecting a cell. RB is present in the cell cycle. RB is a protein that blocks, it's a tumor suppressor, it blocks cell division, correct? It blocks a cell from going and making its DNA and going on with the cell cycle. When people looked at what SV40 did, it makes a protein, which is a viral protein called T, or transformation, transforms a cell into a tumor cell. And what the T protein does is, not surprisingly, it essentially acts like a football blocker. It binds to RB. And when it binds to RB, it takes it out. And when it takes out the RB doesn't work anymore, and so the cell can go ahead and cell division succeeds. So the T protein, the T gene, the part of the gene that codes for the T protein, is called an oncogene. It was one of the first oncogenes, or cancer-causing genes, to be discovered. So the virus brings this gene into the cell, the gene blocks RB, and so the cell can proceed. You'll see it also blocks another tumor suppressor, P53. Another virus was the Rous sarcoma virus, the one that I described discovery earlier on. the Rous sarcoma virus has 3 major genes for reproduction and packaging, and people found there were 2 types of viruses. One of them is the Rous sarcoma virus that causes cancer, and another in the same cell doesn't cause cancer. So now you can compare those 2 viruses, the one that does and the one that doesn't. Oh, look, there's an extra gene. And what is it? It's an oncogene. Because if we take that gene out and we add it to normal cells, they become tumor cells. So people took that gene and they asked the question, they called it SRC, S-R-C. And this was an amazing discovery when it was made. What does SRC do? It turns out SRC is a protein kinase. Remember protein kinases? They're things that add phosphate to target proteins. Change their shape so the protein no longer does its thing or is activated, either way. And it turns out the targets for SRC are a number of tumor suppressors, and also where they block it, and another target for SRC is an oncogene which activates it. So with this background, now we have a very specific description of what's going on. Here is a cell infected with Rous sarcoma virus that has this extra gene called SRC. SRC reacts with the protein, changes its shape, and you get cell division. This was actually one of the first protein kinases to be described in cancer, and now there are literally hundreds. So this is what happens in virus-infected cells. Now let's turn to normal cells, cells that don't have virus infection. We know that 90% of cancer is caused with no viral infection at all. Now, when approaching this problem, we already know about oncogenes, the genes that the tumor virus is bringing in. We can surmise that the oncogene got into the tumor virus by the tumor viruses picking it up. So here's a model for the origin of an oncogene. And the origin of the oncogene is here. So here's a virus genome. There's a host cell gene. And the virus picks it up from the host. The virus essentially picked up this normal gene from the host because viruses don't necessarily have protein kinases. In fact, most viral genomes were originally in the host cells, and they kind of got packaged into a protein. So we would call the gene in the host a proto-oncogene. We'll call the tumor virus one, the oncogene. We'll call the one in the host the proto-oncogene that it picked up. Should the host proto-oncogene be exactly the same sequence as the viral oncogene? No. If you think of evolution and change through time and mutations that happen, there are going to be more mutations in the virus, spontaneous mutations, and viruses don't do repair, and so the gene sequence will be slightly different in the virus than it is in the host. But let's think about this. This assumption, this prediction we're making is that I have in all of my cells a set of genes that, when put into a virus, can cause cancer when the virus brings it into the cell in an active form. And that set of genes is just kind of dormant there, it's just sitting there, and under the right conditions, they could become activated. Well, a couple of scientists and their team, Michael Bishop and Harold Varmus, at the time at University of California, San Francisco School of Medicine, decided to look for the SRC gene from Rous sarcoma virus, the one that's involved that's involved in causing cancer, in uninfected host cells. Now, what was it? Remember, the Rous sarcoma virus infects chickens. So they took chicken DNA. There's 1 billion nucleotides in chicken DNA. And they took the SRC gene, 1,200 nucleotides, and they went fishing, right? They went fishing. They went looking for the SRC sequence by this technique of matching the base pairs in the haystack, right? The needle of the haystack of the huge amount of DNA in the genome of the normal chicken who was uninfected. And lo and behold, they found it. It wasn't exactly the same sequence as the SRC gene in the virus, which you would predict because there's been mutations that have piled up over the time, right? Pretty soon, other viruses were found, viral proto-oncogenes were found in human cells. And so now we have this whole set of proto-oncogenes which now could cause cancer. Both Bishop and Varmus jointly were awarded the Nobel Prize for this discovery. Just a little side issue on Nobel Prizes. Nobel Prizes are, I've mentioned a number of them in the course. Nobel Prizes are given by the Royal Swedish Academy to scientists in a given year who have done major research. But typically they will give it to scientists whose discoveries have led to important things over the years. And so they're not going to give it to a scientist right after they make the discovery. That's why you see Peyton Rous winning it after over 50 years. Bishop and Varmus won it after about 10 years after their major discovery. Nobel Prizes are not given by the will of Alfred Nobel, cannot be given posthumously. That is, a person who has died and made great discoveries in the past cannot win the Nobel Prize. So that's why some of the names that you will hear may not have won the Nobel Prize, and other names haven't won because their discoveries are not deemed as important. Okay, a second way of looking for proto-oncogenes. A second way of looking for proto-oncogenes would be to isolate them from a tumor. So you'll take a tumor that you think has an oncogene in it, a proto-oncogene in it. You take all its DNA out, chop it up into pieces, gene-sized pieces, and now take some normal cells and add pieces to the cells. Okay, and say, piece number 1, do you cause the cells to go into cancer? Piece number 2, etc. And when people did this, they actually isolated cancer-causing genes from normal cells. So the result of these two approaches was that there are cellular homologs of oncogenes, right, in non-infected tumor cells. That is, they are normal cells in the body. So the real challenge here is to figure out what's going on, right? So let me summarize briefly. I've described gene expression, and it's very important to note that I described the various levels of gene expression because the oncogenes that I'm going to describe and the proto-oncogenes I'm going to describe at the molecular level act at those different levels. You recall transcriptional control, and you'll recall number of copies of DNA, and you'll also recall looking at these post-transcriptional things like modifying the proteins, etc. The second aspect was I described the oncogenes that cause cancer that are in tumors and where they got them, tumor viruses, and where they got them by picking them up. And we had various proofs of that. And then we have the evidence that normal cells have proto-oncogenes. Now the challenge is, so we have in our cells genes that can cause cancer if they are taken up in viruses. So what's the difference? How does a gene that's in my cell not cause cancer all the time? What happens to that gene that gets converted? How does a proto-oncogene get converted to something that's active and causing cancer? What do these genes do in viruses to cause cancer at the molecular level? And what do they do in normal cells, normally, where you don't get cancer? And finally, how do you activate this gene in the normal cell with no virus involved and get cancer? And that's the subject of the next lecture. Thank you.